Shani, Ashish.
This meeting is being recorded.
Very good evening to all. I, on behalf of Institute of Pulmocare and Research, cordially welcome you to the 14th session of PULMOCON 2021. The topic today we will discuss is new diagnostic tools in pulmonary practice in the microbiology area. To chair the session, I would like to invite Dr. Bhaskar Narayan Chaudhury. Dr. Chaudhury is a renowned microbiologist. He has published and presented more than 82 papers in national and international journals and conferences. He is an NABL lead assessor, technical assessor in microbiology, serology, molecular testing, flow cytometry for medical laboratories since 2013. He is a life member of IAMM, ISCCM, AHA, CAPP. He is also a member of the Executive Council, Hospital Infection Society, India. Currently, he is associated as a chief microbiologist and senior consultant and infection control officer at Peerless Hospital and Research Center, Kolkata. I welcome you, sir, and I request you to kindly proceed over the meeting. Thank you.
Thank you very much. So good evening, everybody. Thank you, Dr. Bhattacharyya, sir, for giving me this opportunity to chair this session, this very important session on new diagnostic tools in pulmonary practice with focus to microbiology. So we are very fortunate to have one of the most promising microbiologists in our city, I will say, because Shubhranshu, I know Shubhranshu for a long time when he was doing his MD. I know him from that time. So he was briefly in government hospital in West Bengal, then joined CMRI, and now he is in CNCI, Central Government Institution. So he is the best microbiologist to talk on this topic because he has worked with all the different systems, from starting from conventional systems, the automated systems of culture, then now MALDI-TOF, then the different types of other techniques. Now we are blessed to have this PCR technique. So COVID has opened up this molecular diagnostics, which is now available in most of the laboratories of the country, including our city. So not only open RT-PCR, we have different types of closed systems, the multiplex PCR systems, the line probe assay to detect tuberculosis and their sensitivity. So I will introduce Shubhranshu. I'm very fortunate that Shubhranshu has worked with most of these technologies. So Dr. Shubhranshu Mandal is a microbiologist, specialist and in-charge microbiology and molecular biology at Chittaranjan National Cancer Institute. He is posted in Newtown campus. He has completed an antimicrobial stewardship course from University of Dundee. He is a life member of Indian Association of Medical Microbiology, Hospital Infection Society, India. He is a member of European Society of Clinical Microbiology and Infectious Diseases, and he is also a faculty of DNB Microbiology and has more than 11 international publications to his credit. So without wasting further time, so I'm now handing over the microphone to Shubhranshu. So over to you, Shubhranshu. You can start your presentation now.
Thank you, sir. Thank you very much for your kind introduction. So we all, the generation of microbiologists, of our generation of microbiologists, has come to this field of clinical microbiology by seeing you only. So I can only remember that my first job in Kolkata has been given by Sir Bhaskar Narayan Chaudhury. I can always remember that. And thank you, Dr. Parthasarathi Bhattacharyya, for having faith on me to talk on this vast topic. I would like to say that is recent diagnostic methods in respiratory medicine. So without a further ado, I will start my presentation. So the trending topic in the field of biomedicine right now is personalized medicine, also called precision medicine, stratified medicine, or called as P4 medicine. That is predictive, personalized, preventive, and participatory. It is understood as using the right drug for the indicated person at the right time. Although this concept is been gaining more importance in the area of cancer, If we consider all the other specialties, we could say that clinical microbiology, specifically the diagnostic microbiology, is the paradigm of personalized medicine. Several diagnostic methods can be used, ranging from direct methods like staining of organism, like specific gene detection, antigen detection, to indirect methods like serology, in which level of specific antibodies against certain microorganism or antigens are detected.
Shubhranshu, are you changing your slides?
No, sir. No, I'm not changing.
Okay,
I'm not
sorry.
changing. I'm not changing. I'm just giving some pretext.
Okay,
So,
sorry.
So, in general, diagnostic methods must be fast, precise, simple, and affordable. So, however, primary requirement for diagnostic methods are high specificity as well as sensitivity. So, other interesting collateral properties, although it's not essential, would be possibility of being automated and being cost effective as well. For some infection, early diagnosis as well as treatment may have crucial role in curing patients or reducing their mortality as well as morbidity. Since right antibiotic treatment administration at the right time is needed for whatever we are dealing with drug-resistant bug. At present, we are witnessing a significant change in clinical microbiology laboratory led by automation. This trend has been supported by technology like mass spectrometry, as I told, multi-TOF and ESI-TOF. These advances opens the door of greater standardization, operational excellence and performance, as well as better laboratory efficiency. There are several studies demonstrating that changes in diagnostic microbiology reduce the time for generation of test result, which has a clear clinical impact. Despite the fact clinical microbiology laboratories are implementing many advances taking place in our field, if we look towards the future, so other new techniques are on the horizon, including the next generation sequencing. Although it is only used in few laboratories at present, it is undoubtedly one of the methods to keep an eye on. So, if you look into the crisis in the infectious disease scenario right now, we'll say that we are dealing with widespread antimicrobial resistance. We are dealing with XDR and MDR bugs day in and day out in our ICUs. There are increasing number of patients who are immunosuppressed. As I'm working in a cancer hospital, I can say how much of patients are immunosuppressed. Since India is becoming diabetic capital of world, number of patients who are immunosuppressed are being increasing day by day. Emergence of new pathogens. What can you say when you are in the midst of a pandemic of an entirely new pathogen by which we don't know about nothing 2 years ago? Re-emergence of older pathogen. The best example for pulmonary medicine for re-emergence of older pathogen is XDR or PDR tuberculosis. And as you all know, there is drying of pipeline of new drug development. And this bias is due to inappropriate antimicrobial therapy. As we use most of the viral diseases, we use antimicrobial that leads to this biases in our body. So if you look into the historical perspective of the microbiology, for me, these three incidences are landmark in microbiology, diagnostic microbiology. First is in 1880, the cultivation of bacteria by Sir Louis Pasteur. Then the most important method in microbiology, it is gram staining, which is invented by Sir Hans Christian Gram in 1884, and the container for cultivation of bacteria, that is by R.J. Petri in 1887. So what are the traditional methods of infectious disease diagnosis? First is taking clinical history and examining your patient. Then you have to decide whether your patient is actually infected or not, and or any other non-infectious causes is mimicking the patient's infectious nature. So if the patient is infectious or infected, then is it a viral infection or a bacterial infection? Now, by seeing gram stain, you can see whether what kind of bacteria it is, whether it is a gram-positive bacteria or a gram-negative bacteria, or what kind of virus it is. Now, if you want to start a therapy, you need to have a local antibiogram of your hospital, so that you can start the empirical therapy and take after the culture results have come back, and you take a correct treatment decision. So there is a wonderful guideline laid by IDSA in 2018, how to utilize your microbiology laboratory for the diagnosis of infectious disease in 2018. The highlights of this guideline are that microbiologists should have a guts to reject the poor quality sample. So every laboratory should have a sample rejection criteria. Physician should not demand that laboratory report everything that grows in their petri plate. Specimen from that sites such as lower respiratory tract, especially sputum, nasal sinuses, superficial wound, fistula, and other require special care in collection. Whenever needed, you should consult your microbiology laboratory or microbiologist. The laboratory requires a specimen, not a swab of a specimen. Many a times I have a swab from a track. Instead of having a tracheal aspirate, they usually send a swab from trachea. So you should reject that type of sample. A specimen should be collected prior to administration of antibiotic. That is the golden rule. Susceptibility should be done only on clinically significant isolates, not on all isolates that recover in your petri. So specimen must be labeled accurately and completely so that interpretation of result will be reliable and traceable. So we should develop a culture for sending a culture properly and consistently. For that, your nursing team has got a very important role to play. So nursing to ensure safe and timely collection of specimen from appropriate sources, and you need to develop a process that all the cultures that are collected should be properly and promptly transported and labeled correctly and transported to the microbiology laboratory. So how to choose the ideal test? So these are the 5 main pillars for choosing an ideal test. First is the technical efficacy or the diagnostic accuracy or efficacy. You need to understand what are the resources you require for any test to introduce in your laboratory. What is the time to get the results? What is the sensitivity and specificity of that test? And what is the negative and positive predictive value of the test? Then the diagnostic decision efficacy, that how much it is helpful for making a diagnosis or changes the probability in differential diagnosis. Then the therapeutic efficacy, whether your therapy or procedure will change after the test result. Then the patient outcome efficacy, whether the patient is improved with the test, whether to the compare who doesn't have the test done, whether it is having a morbidity and procedure avoided if the test is done, and what is the quality adjusted life year impact. And last but not the least is the societal impact, that is the cost benefit ratio. So as microbiologists are with the lab diagnostician or always have this thing, the race against the turnaround time. Whenever we get a sample, clinician always asks how fast you can give us the results. So there is recent explosion of FDA approved rapid diagnostic test methodology for infectious diseases. It is already been recognized the rapid diagnostic test as well as the biomarker as a key component of antimicrobial stewardship program. There are emerging methods include variety of technology, but they varies in complexity, price, speed, and ability to identify single or multiple pathogens. But the major focus on disease state and pathogen associated with increased morbidity, mortality, and excessive healthcare cost that includes bloodstream infection, respiratory tract infection, and many drug-resistant bugs like MRSA, VRE, ASPL, CRE, so on and so forth. So the old term turnaround time has been now replaced to new term, that is TTI, time to intervention. Multiple studies have shown that if you optimally combine your test result with effective intervention, that will reduce mortality, length of stay, and lower the cost. So you need to intervene, not sitting on the result. If you order a test, if you get that test result, then you need to intervene with your therapy. Next is the point of care test. That is those type of tests that can be done at bedside. These are the original point of care tests. These are the rapid antigen tests, mostly designed for Streptococcus as well as influenza. mostly for influenza in children and adults. These have got a good amount of sensitivity. Now, if you see the FDA-approved antigen-based point-of-care test, you can see BD Veritor, Abbott HD Bioline, your Princeton, Quidel QuickView, Abbott Allier Binax now, Quidel Sophia Influenza AB, Arctium Maria POC recipe. Most of them are based on the methodology of lateral flow immunoassay and give you result within 10 to 15 minutes. Other than Quidel Sophia Influenza AB fluorescent immunoassay, which based on the methodology of fluorescent immunoassay. But again, it gives you a result within 15 minutes. Next is rapid diagnostic tests. So there is lots of FDA approved rapid diagnostic test technology. There are different manufacturers and they have different targets and different targets as well and different time to get the results. First is peptide nucleic acid fluorescent in situ hybridization. This is not available in India. This is a manufacturer is Advanced DX, and by PNA fish you can have a morphokinetic cellular analysis of organism by which you can have a sensitivity of that organism also. There are lots of PCR as well as LAMP platform, that is loop mediated isothermal amplification test, like Cepheid Expert MRSA, BD Max, Meridian Biosciences, Illuminagens, Cepheid Expert difficile, Cepheid Carba R. They all can have different syndrome testing like GI, respiratory, blood. Next is MALDI-TOF MS. I will discuss it in my later slide in details. Two of the companies, Bruker and Biomero, has got this MALDI-TOF MS. But you need to remember that for MALDI-TOF MS identification, you need to have a pure colony. You cannot do it directly from samples. There are multiplex array panel, BioFire film array, which is already available in India, and Verigen and Luminex, which is not available in India. There are other methodology like nuclear magnetic resonance, like T2 biosystem, and which takes into account the blood for their testing. Now, let us see why what IDSA has to say about the rapid viral testing in respiratory pathogen. IDSA recommend rapid viral testing for respiratory pathogen to reduce the use of inappropriate antibiotics. They also suggest rapid diagnostic testing in addition to conventional culture and routine reporting on blood specimen if it is combined with active antimicrobial stewardship program as well as interpretation. That is of utmost importance. You need to have an active antibiotic stewardship program in place in your hospital, and they need to interpret the test result properly. Now, what are the rapid diagnostic tests available? As I have already told, the most common and important rapid diagnostic test in any clinical microbiology till date from 1884 to date is Gram stain. Next is the biomarker of infection or inflammation, that is white blood cell count, ESA, CRP, lactate, procalcitonin. This has been discussed lots of time. I will not go into that details, but I will talk about some new biomarker. First is STREM1, that is soluble triggering receptor expressed on myeloid cell. When bronchoalveolar lavage fluid has been shown to be more accurate than other clinical and laboratory factors in detecting bacterial and fungal pneumonia, when the STRM1 has been evaluated in bowel fluid. The value more than 200 picogram conferred a sensitivity of about 75% and specificity of about 84% in diagnosing pneumonia. There is a single center study where they found a score created from the expression of TRM, that is triggering receptor expressed on myeloid cell 1 on monocyte as well as neutrophil, and the concentration of interleukin 1 beta, interleukin 8 and STRM1 in the lavage fluid were significantly higher in VAP in compared to the non-ventilated patient control. So they have a 7-marker bioscore system, and which can correctly identify almost 89% of VAP cases and 100% of non-VAP cases. There is another interesting biomarker called elastin fiber, which is a marker of lung parenchymal destruction, which can be easily detected in tracheal secretion, and was postulated to differentiate colonization from infection of lung. But unfortunately, it showed very low amount of sensitivity. This may be because this elastin fiber production correlates more with the lung destruction than the infection. Another biomarker is endotoxin measurement in lavage fluid. So whenever you have a gram-negative bacterial pneumonia, you suspect gram-negative bacterial pneumonia, they found a level of 6 EU per ml. Whenever the level is greater than or equal to 6 EU per ml, they distinguish it from gram-negative pneumonia from a colonized patient with pneumonia, or with whoever the patient is having a pneumonia from a gram-positive cocci. There are BL, interleukin 1 beta, interleukin 8, MMP8, MMP9, hydroxyquinone, all were studied. So interleukin B levels has a very high negative predictive value for VAP, and when it is combined with interleukin 8, it had a negative predictive value of 1. So now
We cannot hear anything. Koshi, could you please see?
Connectivity.
This meeting is being recorded.
sir, am I
Yes,
audible
yes.
right now?
Yeah, you are audible.
You are audible.
Yeah. So from when I got disconnected, this Enos from Enos, no?
Yes, he knows.
Okay. Yeah. So there is a very innovative technique called electric nose, where the devices are portable and have a very good potential for point of care application. So generally they use to analyze volatile organic compounds which are being formed by the microorganism metabolites. So in Excel breath, there'd be very good non-invasive and easily applied way to reduce diagnostic time. But sadly, the in vivo and in vitro data varies. But when it can be used as an adjunct alongside other available tests, the probability scores to improve the diagnostic accuracy of VAP. Now, this has been taken forward by multi-center prospective observational study called BreathTX, which is a molecular analysis of exhaled breath as diagnostic test for ventilator-associated pneumonia, where exhaled breath is to be analyzed using thermal desorption gas chromatography mass spectrometry, that is TDGC-MS. Now, again, some non-invasive technique. So whenever we need to check the bacterial etiology, we have to go for either an invasive technique for going for a lavage or mini bell. But there are some pilot studies which are showing very good result, very good PCR analysis result, which are which has a high concordance with bell sample. These are the exhaled breath condensate fluid which are taken from HME filters of ventilators. So this could be a very good alternative for invasive devices, for invasive procedures like bell. So there are other methods like direct e-test. It has been already run. So whenever your patient is in ICU, he or she is critically ill, there are multiple risk factors with multiple comorbidities. they have having a risk of having serious community acquired as well as hospital acquired infection. Those are mostly device associated infection and you need antibiotic, and there is increased mortality as well as morbidity. So the goals of management in the ICU is less mortality, shorter stay, less HAI, and less antibiotic to be used. Serious infection can be caused by virus, bacteria, fungi, or parasites. The present challenges are the current standard of care tests, which depend on culture-based initial diagnosis, often takes at least 48 to 72 hours to provide a result. Culture remains negative even when bacterial or fungal infection are strongly suspected. Viruses and parasites are detected mainly by indirect means, and ineffective antimicrobial therapy, both for bacteria and fungus, increases mortality. And effective therapy requires that the identify the causative pathogen as well as the resistance pattern has to be known. So the need of the hour is reliable direct pathogen identification from variety of samples, syndromic approach for detection, reduced turnaround time for pathogen identification, and ability to predict their resistance mechanism, so that on contrary to have ability to predict the drug susceptibility also. The answer is molecular methods. So molecular diagnosis of sepsis, there are different methods. One is a hybridization technique, that is FISH, amplification technique, that is PCR, multiplex PCR, broad range PCR, post amplification technique like PCR with hybridization, PCR with sequencing, non-nucleic acid tests like proteomics and arrays. So after culture positive, we can do FISH, fluorescent in situ hybridization, Pulmonary or proteomics. From direct samples, we can do a broad range PCR, multiplex PCR, Pulmonary or proteomics. So whenever we have a molecular platform, we can have a specific target, such as tuberculosis in GeneXpert MTB, a range of predefined organisms by use of number of primers, mostly multiplexing, that is in case of biofire Pulmonary. It is clearly important that this panel include all the likely organisms that may be encountered. And broad range assays enabling the assay to detect most microbial DNA, comparing the amplicons against the known libraries. These are sequencing. So if you apply your molecular method to you versus traditional methodology, so if you have a blood culture drawn from a patient and your blood culture flags positive, you do a gram stain, you do a standard identification as well as sensitivity testing, and it requires almost 3 days to start targeted antimicrobial therapy. But on contrary, if you use your rapid molecular method in your practice, you can do, whenever your blood culture flags positive, you do a gram stain and a rapid molecular identification by using a multiplex PCR. You can identify the organism first as well as detect the antimicrobial gene first. Say for example, you have a Staph aureus with make a gene, or a Klebsiella with a NDM positive gene. So from day one, you can start the targeted therapy. Now the syndromic approach. So there are different diseases with common signs and symptoms, but there are different etiologies and that also differ in management strategies. So for that, IDSA has given a example for the exact definition of syndromic testing. The syndromic testing is a symptom-driven broad grouping of probable pathogen into one rapid test that maximizes the chance of getting the right answer in a clinically relevant time frame. That is a very much important thing. So if you see this slide, there are different age groups having similar kind of X-ray picture, but different viral etiology. So there are lots of diagnostic tools available, but these are time consuming, labor intensive, technically complex, often have a very low yield and lacks of sensitivity, specificity. And also there is a overhead cost, limited coverage, confounded by overlapping symptomatology, as well as need to order multiple tests specific for suspected organisms. Sometimes there are unavailability of different organism test is there. So in case of traditional testing, you need to order multiple tests. But in case of syndromic testing, you get a one sample, one comprehensive test, and multiple results in one report. So you need to have a one system which is fully integrated, that is sample preparation, amplification, and detection can be done in single system. So we have right now in our country, we have biofire panels. So we have two respiratory important panel. One is upper respiratory panel, which is now called as respiratory panel 2.1, which has 22 targets. We can get result in 45 minutes, and it has got SARS-CoV-2 in it, as well as influenza H1N1 But it is important to know it doesn't have bird flu, that is avian influenza, in it. So the sample type is nasopharyngeal swab and viral transport medium. So the limit of detection is 160 copies per ml in 2.1 biofire panel 2.1. Targets are M and S gene, and they, as per the literature concerned, they can detect many of the variants. So the detection may not be affected by the mutation of that COVID. So who should get tested for respiratory panel? Mostly young infant, elderly patient, immunocompromised, and patient with comorbidity. Next is pneumonia plus panel, which has 34 targets, and you can have a result within 1 hour. You have 18 bacteria, 8 viruses, and 8 antimicrobial resistance gene. So the sample type is sputum, endotracheal aspirate, bronchoalveolar lavage. Now, the importance of the beauty of this panel, you get a semi-quantitative result. So they have something called bin copies per ml, that is genome copies per ml. So they have created some bins between 10 to the power 3.5 to 10 to the power 4.5, 10 to the power 4.5 to 10 to the power 5.5, 10 to the power 5.5 to 6.5, and so on. Now, according to IDSA recent guideline, you should not report when the gene copies per ml is 10 to the power 4 and below. It has not got clinical significance. So whenever reporting the pneumonia panel, it is very much important to know that below 10 to the power 4 genome copies per ml, and you need to know that this genome copies per ml or bin copies per ml are not equivalent to CFU per ml of the cultures. That is a very, very important part. You need to know that you cannot replicate this bin copies or genome copies with the CFU per ml of that thing. So there is a very, very good study published in Infection and Drug Resistance by ICMR as well as several other corporate hospitals. So they have found that secondary infection in hospital is COVID-19 infection, that is Indian experience, where they have found 3.6% patient develop secondary bacterial infection, and they have got drug-resistant bug like Klebsiella and Acinetobacter in considerable amount of the patient of having secondary infection in COVID. Next is MALDI-TOF MS. So I, being the user of MALDI-TOF MS, I try to describe it as lucidly as possible. So this is matrix-assisted laser desorption ionization time of flight mass spectrometry. This is the entire name of this MALDI-TOF MS. So this process involves laser ionization of molecule. First, you ionize the molecule through laser, then separate it by their charge to mass ratio, and you get a spectrum. This spectrum is matched with the library of the spectrum given for the different organism. If it has got a match, then you have a perfect identification. So you can rapidly identify bacteria, yeast, mold, TB, NTM, but you require pure culture for the identification. That is the most important thing. You cannot do MALDI-TOF MS from direct sample. So there are translational research can be done from MALDI-TOF MS. So so there is a very important theory, very interesting theory, I could say, that most proteins in bacteria, these additions, several virulence factors, not only can trigger immune response, but also aid bacteria to evade the host defense. So identification of these proteins shall be very much helpful to understand the pathogenic mechanism and resistant mechanism so that clinician can optimize the treatment plan to ensure the correct and timely therapy. Moraxella catarrhalis is a very, very important cause of respiratory infection, especially in case of COPD patients. So where outer membrane vesicle has been identified by using MALDI-TOF MS analysis and has gained a very good insight in the biological function of this protein carrying lipid structure, and a novel molecular basis could play the role of outer membrane vesicles in Moraxella catarrhalis pathogenesis. This is a very important new thing which can come up. Now, again, the variation of MALDI-TOF, that is cell-DTOF MS, that is surface enhanced laser desorption ionization time of flight mass spectrometry, which is considered to be an extension of MALDI-TOF, which uses protein chips arrays based on analytical platform, and it has been used for discovery of diseases associated biomarker. Recently, two biomarkers, namely transthyretin and SAA-arginine, was detected by cell-DTOF MS, which in combination with neopterin and C-reactive protein significantly improved the sensitivity and specificity for tuberculosis. So this could be a very important, very nice finding to know. So another advanced technology in the area was about Iridica device, which is a coupling of broad range PCR to electrospray ionization mass spectrum, that is PCR and ESI-MS. That can identify over 1200 pathogens, bacteria, fungi, and it can do it directly from sample. There is no need for culture, and time to get result is almost 6 hours. Radical study, which paired PCR sample with routine microbiology in clinically ill patient, being investigated for potential infection, mostly in respiratory cases. In blood, PCR and ESI-MS has got 3 times more likely to identify pathogen compared to standard blood culture method. And it has a very high negative predictive value, 97%. This PCR ESI-MS could able to help targeted antibiotic therapy in patients who have already commenced on antibiotic and negative culture. This is called salvage microbiology. So many a times we got negative cultures because of prior initiation of antibiotics. So in that cases, PCR ESI-MS could be a very effective tool. Much has been emphasized regarding the importance of rapid initiation of antibiotic therapy in suspected infection, but this must be balanced against the risk and stewardship issues surrounding the over-enthusiastic or inappropriate antibiotic use. So this, if you use MALDI-TOF MS as a part of your antibiotic stewardship program, so there is again reduction in time to identification of bacteria, time to start effective and optimal antibiotic, length of stay reduction, length of hospital stay, reduction in mortality, and as well as reduction in hospital cost per patient. So if you see the landscape of FDA approved diagnostic tests for acute respiratory tract infection, there are some tests called CLIA web test, that is clinical laboratory improvement amendment web test. That is the test where you don't need a skilled laboratory technician to do this test. These are mostly designed by a layman. They can do a test, but without any harm to the patient or without interfering the test result of the patient. These are mostly designed for influenza A, respiratory syncytial virus, and various other multiple viruses. The sample are generally nasopharyngeal swabs and nasal swabs per se. There are some moderate to high complexity assays, but they are mostly designed against influenza A, parainfluenza, respiratory syncytial virus, human metapneumovirus, multiple viruses with atypical bacteria, multiple viruses, bacteria with resistant gene marker. So they give you result within ranging from 15 minutes to 60 hours. So there are some selected nucleic acid based point of care test. As you all know about Cepheid Express Flu RSV, Cepheid Flu RSV XC. There are Biomeru Biofire Film Array Respiratory Panel, Widal Solona Respiratory Viral Panel, Diazorin simplexa TM flu AB RSV panel in Cepheid Expert flu, Luminex Verigen, QIAGEN panel version 2. All of them are based on RT-PCR or PCR technology, and you can get result as early as from 20 minutes to 120 minutes. So as you all know, know about this Abbott ID now, which is a less than 20 minutes point of care test, which has been designed to get influenza AB, RSV, group A streptococcus, as well as COVID. And we use this Abbott ID now, as well as this Acula by Thermo Fisher. When you use a swab to collect sample, place it in the liquid media, pipette it, barcode scan, and reaction container is placed in the instrument, and your result has been ready within 20 minutes. So there are some innovative approaches for future POCTs. First is biosensor, where you can use a gold-coated arrow of carbon electrodes, where the inventor were able to detect Mars-CoV spike protein in picogram range within 20 minutes. This is an electrochemical assay based on the competitive binding of Mars-CoV antibody either to the virus in the sample or to the immobilized antigen in the electrode, and which can be measured by the reduced peak current during the chip. Another sensitive technique is based on the surface plasma resonance, again, which is a biomolecule bound to the metal surface lead to the reduction of reflection of incident light, and it is being done for recombinant avian influenza virus H7N9, and it can have a detection level of few hundred copies per ml in the nasal fluid within 10 minutes of processing time. Again, there is a typical type of PCR that is called capillary convective PCR, that is CCPCR, where the reagent circulate across the temperature gradient in a simple capillary tube and allows round time shorter than 30 minutes. These all are Chinese technologies, and Zhao et al. Integrated this method into a 1.5 kg device for automation for detection of different kind of influenza A viruses. Alternative to nucleic acid based technique, there is a term called GMR, that is giant magnetoresistive biosensor function, which is almost comparable to ELISA, but instead of enzyme, they use magnetic levels. By designing a prototype of lateral flow assay costing approximately $5, Wang et al. Simultaneously detected influenza A and influenza B virus in a swab sample in a very low cost. Now, is the imaging is a proxy to detect infection. All pulmonologists are having written this chest X-ray, ultrasound scan, CT scan. We know that CT scan has got a very important role to play in this COVID pandemic. And ultrasound scan even is a very important tool for diagnosis of pneumonia. It's a bedside tool if you can diagnose pneumonia in ICU. The sensitivity is very much comparable to the normal radiological other procedures. But newer, but a invasive technique where you can directly visualize bacteria and fungi within the alveoli is you need to deliver a microdose of fluorescent smart probes, which can be labeled gram-negative, gram-positive, and aspergillus species. Now, this organism can be visualized using a transbronchial fiber-based endomicroscopy. This smart probes as well as this fiber optic microscope can be delivered through a flexible bronchoscope. It is an invasive technique, but it is a very innovative technique by which the clinician directly can see the bacteria, whether it is a gram-positive, gram-negative, or aspergillus. Next is genomic sequencing. Now, as we all know about a lot of things regarding genomic sequencing, because we are talking about these mutations of COVID, and we know many things about genomic sequencing. So it is increasingly available in the research arena. It's the possibility of metagenomic sequencing. It is a technique that it is able to amplify the entire microbial genome and then sequence it. This could be of use not just to identify the current infection, but for tracking the outbreaks that what we are doing right now, as well as the understanding of the microbiome. As technology progresses, this will change. We'll have the access of more information. we ever had. But the size of technology is also shrinking. As I talk about Iridica system, which take up huge amount of space, it takes almost the entire laboratory. While the new sequencing devices like mini ion, Oxford, nanopore is as small as a smartphone. So you can bring it in the bedside for pathogen detection. So in the next generation sequencing, we sequence a small segment of entire microbial genome and compare the sequences with the established database. For that, pyrosequencing is a first of its kind. Now, next generation techniques can identify and differentiate organism, different serotypes, antibiotic resistance gene. For that, we need a definite database. For that, FDA is in the process of creating a database called FDA Argos, which is FDA database for regulatory grade microbial sequences, which is a regulatory grade database for microbial sequences that would be a reference standard to which NGS generated diagnostic sequences could be compared. Now, direct metagenomics. Metagenomics based assays are among the most promising emerging tools in clinical microbiology, as they can potentially identify any microorganism in any given sample. First is 16S metagenomics. The test was first was invented is sepsis test, which is for detection of microorganism from blood, but gradually it has been drawn from the market because of its low sensitivity, and again it doesn't provide antibiotic sensitivity. Compared to 16S metagenomics, the shotgun metagenomics is based on the untargeted NGS, which reads complete bacterial genomes by massive parallel sequencing and provide precise taxonomical resolution, and also gives the antimicrobial resistance marker. There are two methods available right now. One is ID-Tech DX blood and Carriers NGS plasma. In Carriers NGS plasma, they can identify microcell cell-free DNA from over 1200 microorganisms and showing almost 94% sensitivity in a patient with suspected sepsis. Now this is new. This is host response and transcriptomics. So diagnostics based on differential gene expression in acute infection are of substantial interest. Classifier such as integrated antibiotic decision model and a disease risk score in febrile children is already been talk of the town in America for long. Now, septicide is a test, is first transcript-based infectious disease diagnostics to receive FDA approval in 2017 and continues to undergo external validation to demonstrate its value in different clinical contexts. I would like to say septicide can divide biomarker of infection with the biomarker of the other non-infectious causes that may increase this kind of biomarker. Say, for example, you may have increased CRP in surgery as well as in infection. So this septicide can differentiate between these two things. They can say this is a septic biomarker, this has been rising. So this transcript-based classifier have a potential to characterize patients not only to pathogen, but by the inflammatory phenotype, thereby offering the possibility of successful personalized immunomodulation in sepsis. This is a very, very interesting new thing. So the future state is normally we if you use a positive stain, we use a Malitov, we use a AST setup, then we have our AST in 48 hours or 72 hours. But if you use the genome sequencing, if you can identify the bacteria by Malitov MS and have a DNA extraction and library preparation, we can have real-time resistance gene detection as early as 24 hours. So how can you not tell about TB diagnostic when I'm talking about pulmonary medicine? So as you all know, these upper part is this line probe assay, your gene expert, age as well as expert ultra, this LAMP and TrueLab, this all has been endorsed by WHO for TB detection. Now, what are the emerging technologies which has been from sequencing to GeneXpert Omni, which can be used as a battery operated GeneXpert. You can use at a field setting. There is a 2 days battery backup. You can take it in the field and you can test 2-3 samples at a go. And POC NATs, there are different Chinese companies are making point of care nucleic acid testing and centralized GST. There are, if you want to target large amount of samples, there may be centralized drug sensitivity testing. So these are the WHO endorsed tests: Expert MTB-RIF, Expert MTB-RIF Ultra. There is first line probe assay and second line probe assay. There are loop mediated amplification tests and our own Indian TrueNat MTB Plus as well as TrueNat MTB RIF. So these all can give you result with less than 2 hours time, and these all are endorsed by WHO in different time frame. Now, if you think about the emerging technology, that GeneXpert is coming with Expert XTR cartridge, where we can detect the drug susceptibility of isoniazid, fluoroquinolone, as well as the injectable aminoglycosides. And it has a very good, the prototype version has a very comparable sensitivity with the conventional culture method. But it has got 2 fallacies. One is that you need to change your GeneXpert machine, that is, which is of 6-color machine, to a 10-color version. You need to change that. There is a cost issue for that. And second is that since WHO has been shifting their treatment for MDR and XDR TB management to beta-catecholine, petaminate, and linezolid, and but the developing of highly accurate molecular diagnostic to detect resistance of these drugs is currently impossible due to lack of knowledge on the resistance mechanism of these drugs. So again, we have to rely on the conventional phenotypic method of the resistance detection or sensitivity of these drugs, which are now recommended for the treatment of MDR and XDR TB management by WHO. So you need to know about something about Chinese FDA endorsed molecular test for TB diagnosis and drug susceptibility testing. These are EasyNet, SAT-TB, MELT-PRO, GeneChip, MDR. So this can be done in district level laboratory or reference laboratory. So these are kind of point of care nucleic acid test. And next is a high throughput solution. When you need to test large amount of sample, there are different solutions from different companies which are undergoing WHO evidence evaluation. One is real-time MTB and real-time RIF-RNH by Abbott Molecular. Fluorotype MTB and fluorotype MTB are by Hain Life Sciences, and Max MTB by BD Max. And these are actually multi-disease platform. These platform and also Cobus MTB. So these actually has been employed for other infectious disease diagnosis like HIV, human papilloma virus, hepatitis C viruses, and now they are taking into account for TB testing, for large amount of TB testing. Now the genome sequencing. If you see the genome sequencing strength and weakness, if you see the whole genome sequencing, the strength, in case of whole genome sequencing, the whole genome is sequenced. There is no pre-specified target needed, and it is a comprehensive solution, and it can detect rare mutation as well as heteroresistance. But in case of targeted sequences, which is NGS, where you can sequence directly from sample, large number of genes can be detected. So it is less expensive than whole genome sequencing, detect rare mutation as well as heteroresistance, and it has a very simpler bioinformatics and storage. The weakness of whole genome sequencing, that it requires culture isolation. You cannot do it from direct sample, and it is a bit slower and having a complicated bioinformatics system. And for next generation sequencing and targeted sequencing, you need a knowledge of target. You need a knowledge of target which to be sequenced, and you get a less information than whole genome sequencing. Both of them as of now is expensive. So if you see the schematic diagram, if you have a sputum sample, you need to extract the DNA, then prepare a library and do a targeted sequencing, then do a data analysis and interpretation, and you get a result for the sensitivity of TB. Now is a total laboratory automation. So the idea of total laboratory automation has been there for long in other segments of laboratory like clinical hematology, clinical biochemistry. But the large reason for not having total laboratory automation in microbiology is the processing of variety of samples, which can be processed in microbiology lab. So this variety has prevented a one-size-fits-all approach to sample processing and distribution of culture, which would be cornerstone for automation. But right now, two of the system having total laboratory automation in clinical microbiology, such as BD Kiestra and Copan Diagnostic WASP Lab, which has been now taken by the Biomiro. So I had the opportunity to see BD Kiestra while doing my observership in ECMED. So it has a wonderful system, but the problem is that the cost and the size. You need a huge space, humongous space for having this total laboratory automation for microbiology. So if you see the entire circle as proposed by WHO, the diagnostic stewardship as well as the antimicrobial stewardship, diagnostic stewardship is to choose the right test for the right patient at the right time, and antimicrobial stewardship team has to rightly interpret the test, choose the right antibacterial and in the right time to make the diagnosis and treatment part complete. So nevertheless, the respiratory tract, there is always constant concern whether the pathogen is colonizing or infecting. The one advantage of culture over molecular technique is the ability to quantify microbes. Though not universally agreed, a cutoff of more than 10⁴ coloniformic unit per ml is often used to identify infection over colonization, though it is not 100% foolproof. I am again telling. So key takeaways will be appropriate indication and specimen collection is critical for both basic microbiology as well as newer diagnostics. No one rapid diagnostic platform meets all. So you have to select tests based on the workflow and the patient population you are catering. Rapid diagnostic can definitely decrease the diagnostic uncertainty. To be effective, rapid diagnostic have to be actionable and tied to the local stewardship program of the hospital. Monitor, you need to monitor for the unintended consequences. Testing must be correlated with overall clinical condition of the patient. And to conclude, new diagnostic tests should be evaluated as to whether they are value added or not. How will the detection of certain resistant mechanism affect our choice in the antibiotic therapy? You need to know that. It is important to establish a collaborative guidance within the clinic with the clinician upfront, involving multiple stakeholders, like what we are doing right now with appropriate education. You need to educate the clinician. What are the new tests available? What are the alternate tests available? What is the turnaround time? What are the information you can get it from the test? There is important to communicate between the antimicrobial stewardship team as well as rapid diagnostic and for improved process and outcome. And who and when get notified when an organism and resistant marker is identified by rapid diagnosis. In my hospital, I create a simple WhatsApp group. I just give them information in the WhatsApp group so that everybody get informed. So while being widely available, RDT remains costly. The clinical demand and appropriate infrastructure are necessary for healthcare to realize the return on investment, to realize the value proposition. So clinical microbiology are going through a very exciting changes. New technologies and vast improvement to existing technology are rapidly improving patient care and decreasing the patient cost through reduction in time it takes to identify infectious agent causing the disease. Currently, diagnostic microbiology seems to be at a crossroad between a conventional testing and implementation of advanced testing platform and full-scale automation. Admittedly, there are concerns around the explosion of this advanced technology because laboratory you need to scrutinize that and decide what works best for the laboratory environment, because a small rural hospital is not likely to have maybe have a fully functional lab. It's not likely to have that amount of volume as a larger hospital in an urban center. Therefore, rural locally would likely not benefit greatly by moving a fully automated laboratory or incorporating a bulk of current and future diagnostic technology. There is also a concern that new testing panel with all their advantages will lure physicians into ordering tests that are unnecessary and therefore potentially increase the patient as well as laboratory cost. On top of this, there is the issue that so much information is being produced from this test that interpretation of actual meaning of the result could be made more difficult and altogether lost in translation. This could potentially lead to poorer patient outcome as well as reversion to conventional method again to provide answer that the panel could not answer. So these are the issues are to be taken into consideration when incorporating a new technology into the existing laboratory workflow. Thank you so much for your patient hearing. I am ready to answer any questions.
Okay, thank you very much, Shubhranshu, for a very exhaustive talk and very elaborative, I think a very illustrious and excellent talk, I will tell. I was thinking that I am sitting in the medical college in the third year and listening or as a PGT and listening to a professor talking. Okay, so it was very mesmerizing. So thank you very much. You have covered everything, I think, from A to Z, everything you have covered. So
Thank you so
I
much.
hardly have any comments to make. So I will open the session for questions. Any questions? I will request Dr. Parthasarathi Bhattacharyya sir to speak something.
No, there are a few questions in the chat box. Yeah.
Okay, so I can't see any. Only one question I'm seeing.
Okay. Let me read it out. Extremely nice presentation. COVID has really played a key role in recognizing the contributions of our microbiology colleagues amongst lay people in general and the medical fraternity in particular, which was due to them. This is an appreciation of the lecture. And the other thing is, other question or other comment is biofire technology is it available and cost is prohibitive or not?
Yeah. Definitely, cost is a prohibitive thing. That I have already discussed because cost is a big issue. Many of the open system platform has got multiplexing can be done on that open system, so that this cost issue can be alleviated, and similar sensitivity can be done also by open system platform by doing multiplexing in open system platform.
What I have a small question.
Yes, sir. Yes, sir.
There is n number of bacteria, n number of fungi, n number of viri, and there's a whole lot of microbes. But to know clinically relevant infecting organisms, like in bacteria, we look at the colony count. Is it possible that you have a correlationship with your what you called as bit copies or
bin copies? Yeah.
Copies.
There is a definite guideline from IDSA. Up to 2020, there is no definite guideline for multiplex PCR for which semi-quantitative result could be clinically relevant. Now, in 2020, there is an article in Journal of Clinical Microbiology in 2020 October, as far as I remember. They have denoted there are two clinical vignettes given for that this kind of problem could arise. Because sometimes you may get different kind of bacteria, 3-4 bacteria, 3-4 viruses. So how a clinician know which is the actual pathogen? And again, the problem of this film array pneumonia panel is that if you have multiple bacteria and multiple genes, multiple resistance marker gene, you cannot relate that which bacteria is having which gene. So if you have, say for example, if you have a Klebsiella as well as Pseudomonas, and you get CTXM, NDM as well as OXA, you cannot relate which has what. Number one problem is that. Again, you have to believe on because only having the gene is not the what do I say, that the expression of gene is much more important. The bacteria carrying the gene doesn't mean it will express it. So for that, we need to follow the sensitivity also. That is much more important. And now talking about the bean copies, this has been clearly noted. If you see, in my talk also I told that 10 to the power 4 bean copies, that is 10 to the power 4 genome copies per ml, should not be reported. It has been clearly written in that article. 10 to the power 4 bean copies should not be reported. Beyond 10 to the power 4, that is 10 to the power 5 and above, you can report it.
Yes, I have seen from my clinical experience from my experience in the lab that actually less than 10 to the power 6 doesn't grow on culture.
Exactly. Sir.
Okay,
that is
copies.
also
So
that is also my thing.
yes,
Also.
and
Even,
sometimes we get lots of these resistance genes, but not all of these are expressed. So
exactly.
it should be used in conjunction with culture.
Culture, absolutely. These two things go hand in hand. These two things go hand in hand.
Another most important thing is that in culture you can know the sensitivity pattern. Where in
exactly
such methods you just detect the bacteria or the fungi or the organism, but you cannot really say to which all drugs it is sensitive to.
No,
Sir,
actually we can also detect the resistance genes
can
in
be
some
possible,
of the panels. In lower respiratory we can detect the resistance genes also. So that so if it is a carbapenem-S producer, we can detect the carbapenem-S. So it's better not to give carbapenems to those patients. So we can have an idea of that also from two panels. One is BCID panel, one is the pneumonia panel.
Pneumonia panel. Yeah.
I think that's a good information that you can have.
And that is another very important thing, sir, that whatever molecular platform will come in future or is there in now, culture still remain the gold standard. Remain the gold standard.
So there is one more comment or question. Yeah, no, they're the same thing. What is hurdle for making multi-top more popular and available? Is it only the cost?
Is mainly the cost. It's mainly the cost because, sir, one experience I can share with you. When I was doing my observership in Europe, so in ID, so some of them asked him, how you identify your organism? So I told them that we use Vitek ID and AST. So they are quite surprised because you are having so much money for identification, because a ID card will cost 250 rupees. And for multi-top, it will cost you 22 rupees. The main problem of multi-top is the capital investment. You have to invest 1.5 CR upfront. But after that, the running cost is very low.
Yes, that's true.
But standing as a clinician, what I feel that there should be very comprehensive courses to get ourselves acquainted to this latest developments, and there should be step care sort of system so that step by step clinicians can get acquainted to. And another problem is that out of so many tests, which one is the fitting test for a particular circumstances and a particular scenario, even a particular clinical scenario, that has to be defined and guided thoroughly to the clinicians. Otherwise, we may be lost in bewilderment in future.
Exactly. That is the most important thing, sir. The communication as an educational program. That is the main thing. That what are the tests available in your laboratory? What are the limitations of the test? What are the alternative of that test? What are the cost of that test? And what is the turnaround time? And what are the amount of information you are getting from the test? You need to educate the clinician about this thing.
True.
So there is no more question. There is thanks from the rapid test for fungi. Is there any?
Yeah. Actually, rapid test. Actually, in CNS panel we have this Cryptococcus for in biofire. But other than that, the problem with fungi, you see, Sir is doing Aspergillus galactomannan for long in PLS, as I can remember, because I used to send Sir, because we still depend on biomarkers, because the standardization of PCR is very difficult in cases of fungi. That is the main problem with fungi, because many of them are coming up, because in this pneumonia panel also they don't have any fungus.
But what about the
We have
atypical
PCR for Aspergillus and
Aspergillus
Pulmocare,
PCR.
and now PCR takes a very short time because all the labs have automated extractors. The extraction time has been reduced a lot, and the analyzers are also being developed with a run time. So we can give PCR reports within 4 hours, say 3 hours or 4 hours.
Exactly.
So PCR is also a very rapid technology that has evolved, thanks to COVID for that. Okay.
For us, one problem is the NTM infection, non-tubercular microbacterium.
Yeah. Sir, this is my favorite topic, NTM. So it's my favorite topic, sir. NTM, if you see.
You spoke about line probe assay. Line probe assay is a very good.
So, sir, there are two methodology. What I worked for last 7-8 years. One is line probe assay. So we have, sir, two DNB students doing thesis on the non-tubercular mycobacteria. One is the incidences of non-tubercular mycobacteria among the pulmonary samples, as well as the extrapulmonary samples, and another one is doing the sensitivity AST pattern of NTMs. So I have done two of this. I have guided two of these topics. So and the incidence of rapid growers, especially the fortuitum and SSS, is very much high in our context. Whatever culture we have done in our setting. And another very important tool will be multitop, because line probe assay again will need a trained technician, as well as this 2 incubator, 3 different rooms. But in case of multitop, if you grow the sample in midget tube and you send and you define that this is not TB, this is MTM, even it is TB, you can send it for multitop, and we can readily differentiate between different other species of non-tubercular mycobacteria. Generally, in line probe assay, the common pattern is we use 2 types of cards. One is CM card, that is common mycobacterial card, which includes TB as well as the non-tubercular mycobacteria, and another card is ACE card, that is additional species. In this CM card, as far as I remember, you can get 14 mycobacterium, including the Mycobacterium tuberculosis. And this ACE card is some atypical non-tubercular mycobacteria, where you can have some additional species. Now, for multitop, you can have comprehensive solution. You can have all NTMs.
It is rightly commented by Dr. Sunny George that multitop is in NTM is exciting.
Yeah.
So seems there is no more question. Gold standard to confirmation, I think he meant. This is Devinder Singh Dhandwal. Yeah.
Gold standard for confirmation of what? NTM?
Possibly is meaning that.
Okay. Gold standard for confirmation of NTM is culture. Obviously, culture followed by molecular methods or multitop, or because after culturing, you need to speciate that NTM. So after culturing, either there are some PCRs other than this Hain test, that is line probe assay, which can say this is TB or NTM, but cannot differentiate between NTM. For differentiating NTM, you need to do this line probe assay or else multitop.
Okay.
Yes.
So Bhaskar, we think we have to wrap it up today.
Yes, yes. Okay. Thank you. Thank you very much, Shubhranshu,
Thank you,
for a
sir.
very enlightening talk.
Very wonderful.
And thank you, Dr. Parthoda, for giving this opportunity
it's
to be in that
my
platform.
pleasure that you have agreed, and I will be. I am grateful to you. I'll rather be after both of you in future, so that we can learn a little bit.
Okay.
Thank
Okay.
you, sir.
Thank you.
Thank you. Thank you, sir. Thank
you very
you.
much.
Thank you so much.
So, with
And
this to
thanks
all,
to the audience also.
Thanks,
The
thanks to the
questions were
patient
very
audience.
nice. Okay.
yeah, thank you. I again express my heartfelt thanks to both of you and to the audience. This is the last module of our PulmoCon 2021. It was incidentally extended to 2022, but we will continue to have a different academic program on a web platform. We'll have regular postgraduate classes and other programs. So it will be my pleasure to send you messages and information regarding those programs, and I would love to see you again and again to our online platform in different programs in future. So with that all, I thank you everyone with folded hands and hope to see you once again. Be safe and keep health.
Thank you,
Remain
sir.
healthy.
Thank you. Thank you.
Thank you.
Good night.
Good
Good night,
night.
sir. Good night. Good night.